The Otto Warburg Medal 2020 will be given to Patrick Cramer, the Max Planck Institute for Biophysical Chemistry in Göttingen. As you see, this is the award 2020, planned for the Mosbach meeting a year ago, as Blanche has already pointed out, that had to be first postponed and then cancelled. The award 2021 will go to Petra Schwille and will be handed over in Düsseldorf this year in fall, also in a virtual manner, so we are experienced with virtual meetings. And in 2019, Marina Rodnina was awarded the last one live in Tübingen. So I think you can see it's a great event now, and we are trained to virtual meetings. But of course, this is the award of the GBM in that we celebrate since 1963. And the GBM decided to provide this award on the occasion of the 80th birthday of Otto Warburg, a great biochemist in the last century, who was awarded with a Nobel Prize in 1931 on the discovery of the nature and mode of action of the respiratory enzymes. So Otto Warburg Medal is intended to commemorate Warburg's outstanding achievement awarded by the German Society for Biochemistry and Molecular Biology, and the prize honors encourages pioneering achievements in fundamental biochemical and molecular biology research. And when we go through the nomination and see, I think it's absolutely clear that Patrick Cramer truly devotes this award because he has in fact truly contributed to fundamental biochemical and molecular biology research. But let me start with introducing him to you. Patrick studied chemistry in Stuttgart and Heidelberg, with internships in Bristol and Cambridge. He chose great places, as you can see here from the photo. He performed his doctoral thesis at Christoph Müller's lab in Grenoble at the EMBL with great success. Then he went to Stanford on a DFG scholarship, where he began working with Roger Konberg on the structure of RNA polymerase. Crystals of RNA polymerase had existed in the lab for some time, but they could not be used for high resolution structure. And with help of novel techniques, Patrick succeeded in dramatically improving the solution and solving the structure of the multi-enzyme complex. Not least for this work, Konberg was awarded with the Nobel Prize in Chemistry in 2006. After returning to Germany, he immediately received a tenure-track professorship at the LMU in Munich, where he stayed from 2001 to 2014. Since 2014, he is director of the Max Planck Institute of Biophysical Chemistry in Göttingen. Patrick Cramer and his group have succeeded in crystallizing RNA polymerases not only alone, but also in complexes with short DNA strands and in determining the structure of these highly complex molecular machines with high resolution. This enables completely new insights into the various phases of the reading process. For example, in other words, the individual steps involved in transcribing the information in the DNA into the information in the messenger RNA. This work is a milestone in biochemical structural research, comparable to the elucidation of the ribosome and the use of the information of mRNA in protein biosynthesis, which we all currently importantly know or learned from. After the pioneering work on the structure and function of RNA polymerase II, Patrick expanded his work to structure elucidation of RNA polymerases 1 and 3, ribosomal RNA and transfer RNAs. All of his work deals with very large multi-protein complexes the production of which is very time-consuming and requires the development of new technologies, and it's so competitively that it's only established in very few laboratories all over the world. Studying the interaction of these complexes with RNA and DNA is challenging for biochemists and structural biologists, but these studies are routinely carried out in Patrick Cramer's lab, as we can see from the great outcome of his work, and we will hear more about that in his lecture soon, where he shows his latest data. He is a world leader in this field, and his work has received the utmost attention within molecular life science. Patrick Cramer's work is an outstanding example of the value of detailed biophysical investigations of important biological processes. In this case, one of the most important complex machineries in biology. His dedicated application of exact methods has added enormous and essential detail to our basic understanding of RNA machineries. They also illustrate that the full power of the amazing structural methods available today can only be realized with the proper applied principles of physical biochemistry. However, Patrick does not only stick to structure, he wants to expand the structure into function. Thus, he addresses the complex regulation of gene transcription, for example, by indirect promoter recognition. Patrick's work combines structural methods such as X-ray crystallography and electron microscopy, but also with methods of bioinformatics and functional genomics. Thus, Patrick expands the research area, the life sciences, and contributed to the development of genome biology, social system biology, and the this talk entitled today RNA polymerases and the COVID-19 pandemic will let us know. Patrick's work has been recognized by a number of prizes and awards. This includes the Leibniz Prize of the DFG, 2 ERC grants in 2010 and 2016, the Century Award of the British Biochemical Society, and this is just to name a very few of them, and most recently in 2021, the Louis Chantet Prize for Medicine and the Hector Science Prize. He's a member of the National Academy of Science of the United States, of EMBO, and the German National Academy of Science, Leopoldina. Thus, it is a great privilege for us to add the Otto Warburg Medal to this list of honors. Thanks a lot. And we are looking now forward to your presentation, Patrick.
Just a very short interruption, I'm sorry. We have the sponsor Elsevier and BBA, who is sponsor of the prize money of the Warburg Award. And we have a very, very short video of Joe D'Angelo. He's executive publisher of Elsevier BBA. And I just want to share the screen briefly with that video. And after that, we will go to Patrick's talk, if this is okay. Mm-hmm.
I'm Joe D'Angelo, and I'm an executive publisher at Elsevier, and I work on the Biochemica at Biophysica Acta journals, or BBA. So Elsevier and BBA are proud to be able to continue our long partnership with the GBM to support the Otto Warburg medals. We're also delighted to be able to extend our congratulations to Professor Patrick Cramer, the recipient of the 2020 Otto Warburg Medal, who's being recognised for his groundbreaking work on gene transcription. I hope that you'll all join me in offering your congratulations to Patrick Cramer, and I hope those of you who are able to attend the virtual ceremony enjoy the talks. Once again, congratulations, Professor Cramer.
Well, first of all, thank you so much. Thank you, Annette, for the very kind introduction and the flattering laudatio, and thanks to Blanche Schwabbach also for the introduction, the organization, and of course also to BBA for sponsoring this award. Receiving this medal gives me great pleasure, but it is also a special honor for me to receive it. And that is because, first of all, it's given to me by close colleagues, so colleagues who are active in research and teaching in biochemistry and molecular biology all over Germany. And I admire them and I thank them for all the work that they do for our field. And second, it gives me special honor because amongst the past awardees, I found a lot of colleagues who I highly value, and many of them also being role models for myself. Just to give you an example, in that list, there's also Max Perutz. And I will never forget when I was a student at Cambridge in 1994, he would come to the laboratory, ask questions, give me advice. And this was just after his 80th birthday. So you can imagine that for a young student, seeing the founding father of the field of structural biology talking to you directly on the bench, that was something very special and probably decisive for my career in structural biology. I wanted to also say that I accept this medal on behalf of the many past and present co-workers, and I would like to thank them for the great contributions that they have been giving to our research over the decades. The medal also provides an incentive for us to stay at the forefront and to break new ground and to shift the boundary of knowledge in molecular biology. This is what we want to do over the next years. We all, however, stand on the shoulder of giants, and that's why I would like to thank also the international scientific community in our field. Without the many great colleagues, it would not be possible to advance and to bring about all these scientific results. And finally, I would like to thank the taxpayer, because what we do is actually very expensive, and I have been generously funded throughout my career, and I'm very grateful for this trust that the taxpayer provides to us. And of course, last but not least, I would like to thank my wife, Stephanie, our two children, Leah and Dominic, as well as the whole family who has been always extremely supportive over the last 3 decades to make this possible. Now I would like to start with the actual presentation. And for this, show you the first slide. Which actually lists all the past and present co-workers. And you see many young people have joined us over the years from, I think, around 30 or so different nationalities. I must say that this is probably one of the greatest pleasures in our job, that we not only are able to see things that others have never seen before because we embark into the unknown, but also that we can work with all these young people who are from very different backgrounds, and you learn a lot by interacting with them. Now, today, as was said, I will not provide an overview of what happened over all these years, but rather present really the most recent research. And some of it is actually unpublished data. And what I was thinking is to summarize how our research on RNA polymerases has an impact on the coronavirus pandemic, or probably for now, better to say how it's related to various aspects of the ongoing pandemic. As you know, RNA polymerases are the central enzymes that make long chain-like RNA molecules, and they have a central role in all living processes in cells, but they're of course also important for viruses and bacteria and so forth. So, when the pandemic was striking Europe maybe a year ago, of course, we were first of all totally shocked. We all went to home office and took all the safety measures. And then we had our first Zoom conference, probably a year ago. And we thought, what could we do? We were all a bit puzzled about the situation. Everybody had to stop their projects or move to evaluation of data sets and so forth. And we thought, how could we help with the pandemic? And of course, we are not drug makers. We are not people who can make a vaccine or anything like that. But we thought, we are experts in polymerases. And so what can we do with our knowledge and our expertise and the toolbox that we have in order to help with the ongoing pandemic? And so it was quite easy to decide that we should go after the coronavirus polymerase. Coronaviruses encode for 29 proteins. One of them is the polymerase, and it has accessory factors. So it's arguably the central enzyme that is central for the life cycle of the virus, because it is replicating the viral RNA genome and also giving rise to smaller RNA fragments that are important for the viral life cycle. So this enzyme is an RNA-dependent RNA polymerase. And not only is it important for the life cycle of the virus, it's also a central target for drugs that aim at fighting the virus. And when we then looked through the literature, we found that many antivirals are actually polymerase inhibitors. So if you go to hepatitis C, for example, also HIV or the herpes virus, there's medication available, and often this medication involves antiviral substances which target the polymerase of these viruses and inhibit that polymerase, which is actually doing different kinds of jobs depending on the type of virus. And as you know from the media, for sure, remdesivir was the first approved drug against COVID-19. It's not very effective, but it was the first one, and it was heavily used in the clinic, and is a polymerase inhibitor. And now there's new substances being studied, for example, molnupiravir, which is currently has passed phase 2 trials. So it went into patients, and the results are relatively promising, as we can read. Now, we were very encouraged, polymerase is the central enzyme for the life cycle, but it's also a drug target. So then we said, okay, a few of us can go back to the lab, and this team of experts with different expertise then, in record time was able to make active coronavirus polymerase and to solve its structure. So here you see the minimal assay that we quickly developed, where you have a short RNA hairpin, and then you make a recombinant 3-subunit polymerase, which consists of the catalytic subunit NSP12 and 2 accessory subunits NSP8 and NSP7. And as you can see, this enzyme can elongate the RNA primer that you provide quite efficiently. So now we could go ahead and try to solve the structure of this enzyme. And we use cryo-electron microscopy. Here you see the inside of one of these high-end, about 4-meter high cryo-electron microscopes. And with this, you take millions of images of particles of the polymerase. And then by sorting and averaging these particles, you can reconstruct a 3-dimensional density map, which then allows you to build an atomic model of the enzyme. I should say that, this looks all simple now, but it was actually quite difficult even for very, well-trained people who worked on it. And that is because the coronavirus polymerase is actually a relatively small enzyme. And it's only very recently that cryo-EM is able to resolve atomic structures of such small enzymes. And we had other problems. For example, we had preferred orientation of the particles. We could only see the enzyme from one side, but not the other. So actually, we had to use our entire toolbox and also some serendipity to eventually solve the structure. And that was, after many races in my career, this was the most dramatic race we ever did. It was less than 2 months from ordering the genes, the DNA, to clone the polymerase to the publication online, when we had our structure written up and posted on the internet and distributed by Twitter to the international community. And actually the same months, 3 other research teams have solved the same structure, and all of them were located in China. So you can imagine they were under enormous pressure there. and probably also started in time realizing how problematic the situation is. But so we can now compare these 4 structures, and they're virtually identical. We see some additional domains that I will talk about, but otherwise this shows, you know, no matter where you do such science around the world, if you do it rigorously, you come to the same result. So here's the structure of the coronavirus polymerase, just very briefly. The large subunit is NSP12, and it's shown here in different colors. This is the catalytic subunit that also holds the active site of the polymerase, which is symbolized here by this magenta sphere in the center. And then we have 2 subunits of the accessory subunit NSP8 and 1 NSP7, which is this blue molecule here. NSP8 are the 2 long green proteins on the top that actually touch the RNA duplex. So you see in blue is the RNA template strand. That is basically a fragment of the coronavirus genome that is going to be copied. And in red, you see the product strand. That is the RNA that is being synthesized by the polymerase based on the template sequence. So we could make the very first movie. It's a very simple movie, maybe almost oversimplified, but it gives you an idea of how the coronavirus basic replication reaction works. So the nucleoside triphosphate substrates are entering here from the right. They bind to the active site of the enzyme, and then a phosphodiester bond is formed at the 3' end of the growing RNA product to extend the chain, and then the enzyme translocates to the next nucleic acid position, and the whole nucleotide addition cycle can start again. Now, what is special about this coronavirus polymerase, or is it actually just one of many polymerases and there's nothing to be learned? Well, it was actually quite surprising that there is something to be learned, because we do have from the last decade several structures of other viral polymerases, from hepatitis C, from poliovirus, norovirus, and so forth. And these enzymes all have similar active sites. So around this region here, where catalysis takes place, you have a similar structure. And that actually explains why many of those antiviral compounds that I showed you would actually also work against other viruses, because they can also target related viral polymerases. But then look at the coronavirus polymerase. It has, in addition, these long extensions here along the exiting RNA duplex. And these are actually enzyme elements that we call the sliding poles. And they have been shown by others in the community a few years back to be important for the processivity of the enzyme. So what do we mean by processivity? It's actually the property of the polymerase to stay attached to its RNA template until the replication process is completed. And this is especially important for the coronavirus, because it has the longest known RNA genome, about 30,000 nucleotides in length, the longest one that we know. And so it's probably especially important for the coronavirus to stay attached to the RNA, not to fall off during replication. And that's our interpretation for why this polymerase has evolved these sliding poles that you don't find in other polymerases. Now, what is interesting is that this polymerase, although it's small, it can do several different jobs. So it's not only replicating the genome, but it's also producing a set of nested so-called subgenomic RNAs. So these are copies of the genome that don't have the full length, but are actually shorter fragments of the genome. And what is really intriguing here is that these are not just fragments from one end, but these are fragments that contain both ends correctly, but then those ends are somehow fused in a way that we don't understand well. So there is portions in the middle of the coronavirus genome that are not copied. And so in the field, it was thought that there is what is called a template switching step. So the polymerase would start to replicate, and then it would switch to a downstream position on the genome, and that could be located even 25,000 nucleotides away. And this is a very enigmatic discontinuous process that has also been called transcription in the field. And we didn't know, how that could possibly work on the molecular level. It's known that there are certain sequences involved. So you have a transcription regulatory sequence in the genome, and then the polymerase was thought to jump from one sequence to another one, which is very far away. So how should that possibly work? Well, we don't know either, but I want to show you today an unpublished model, which I think at least provides a possible explanation for how this may work. And, as often in science, you have to expect the unexpected and look for serendipity, and not just get lucky, but also to accept the luck that is offered by you by nature. So what we saw in our data is sometimes evidence that polymerase can form dimers. So you see here always two polymerases associated with each other in these 2D classes of these particle images. So we wrote a little script to extract those from the data, and then we solved a new structure, a new form of the polymerase, which is actually an N-type parallel dimer. So here the polymerases would travel in opposite direction, but they hold on to each other in a new type of enzyme with two activities. So what we think, and that is our speculation, it's a model, is that this can perhaps explain better how such shorter subgenomic RNAs are produced, because you could imagine that one polymerase backtracks and then it would expose the 3' end of the RNA, which contains the complementary sequence to the transcription regulatory sequence. And that would then be loaded together with a second site on the template strand with which it hybridizes. It would be loaded into the second polymerase. So our idea is that it's not the polymerase that switches to a new template position, but it is rather the RNA template together with the product RNA that switches to a second polymerase. So it's not template switching, it's polymerase switching. That is our proposal. And of course we have to prove that, but at least, we have now a model which we can follow up. Now, I told you it's the major drug or one of the major drug targets in the coronavirus life cycle, this polymerase. So what can we learn about the mechanism of action of this remdesivir drug that is in the clinic? Well, first of all, you need to look at it biochemically to study mechanism and then structurally. So it was long known that remdesivir can, when it enters the cell, it's a prodrug which then is metabolized to give rise to this triphosphate form, which is the active form of the drug. And that actually mimics ATP. It's very similar. It just has here, this CN group, this nitrile group, and a few little differences in the structure. But the CN group on the ribose, as you will see, is important for the mechanism of this remdesivir drug, how it inhibits the polymerase. So it can mimic ATP, and it can be incorporated into growing RNA instead of the AMP moiety. And we could see that easily in biochemical assays. So here you see the extension of an RNA when you have all 4 natural nucleoside triphosphate substrates present. You get extension to this position +11 as we have designed it to be. So you add these 11 nucleotides, no problem. But then you replace the ATP by remdesivir triphosphate, the active form of the drug. And then what happens is the polymerase still elongates. It's not totally blocked, right? It still elongates to the end to position 11, but there is this elongation barrier encountered exactly after the addition of 3 more nucleotides. So a total of 4 nucleotides are added. First remdesivir, and then 3 more nucleotides. And then you see the polymerase is pausing, it's stalling, but it's not terminating replication. If you wait long enough, or if you have high enough substrate concentrations, then you will elongate the RNA to the end. So now what we have to find out is, why does the polymerase stop here exactly after 4 steps? And that is summarized in our next movie. I cannot show you all the details, but basically the way we do it is like in the good old days when people were making movies for cinema first. You have various snapshots, right? And then you can animate a motion by putting these structural snapshots together. And this was only possible in a collaboration with Claudia Höbartner's lab at Würzburg. They are RNA chemists, and they could make RNAs that contain remdesivir, which we then structurally or used for our structural analysis. And that is actually how the movie that I will show you now was possible to obtain. So here you see a movie of what happens to the coronavirus polymerase when the drug remdesivir enters. So it will be incorporated into the growing RNA. And you see this ball here, this is the CN group, 2 atoms that are there in addition. But the polymerase doesn't stop. It can now incorporate 3 more nucleotides. So RNA chain elongation is continuing. But then after these 3 more steps, you see there's a steric clash here of the nitrile group on remdesivir with a serine side chain in the polymerase active center. And this steric clash, which is really a mild clash, gives rise to this elongation barrier. But you know, simply by thermal motion, the enzyme eventually will overcome this barrier. So remdesivir is actually not a very good inhibitor of coronavirus replication. It's just causing some delay in the elongation, but eventually the process can continue. So that means we have to look for better inhibitors, right? Ideally, we would look for totally new inhibitors that could be further developed one day into new drugs, hopefully against coronaviruses. And how do you do that? Well, the idea is that, you know, these antivirals that I described so far, which are on the market, these are all nucleoside analoga. So this means that their mechanism of action always involves a mimicry of the natural substrate NTPs and an incorporation into the growing RNA. And then, you know, many of them would stop the polymerase immediately, but others would stop the polymerase later or would not stop it at all, maybe just pause it, as we see for remdesivir. Now, the issue why these antivirals that are on this nucleoside analog basis, why they don't work for coronaviruses, is that coronaviruses have evolved a strong proofreading activity. So they contain an exonuclease that can digest away the RNA 3' end after such a misincorporation has occurred. So when the drug enters and it's added to the end of the RNA chain, the polymerase realizes this, and there's an exonuclease being recruited, and the virus actually corrects that error, and then it can go on as if there would be nothing. So those antivirals are not effective. And so I think what we have to look for is something entirely new, a molecule that can inhibit the coronavirus polymerase with a totally new mechanism. I would call it an allosteric inhibitor, which binds to a different site on the enzyme and blocks the enzyme function. And how can you find such a new molecule? Well, you have to go back and screen, you know, large libraries of chemical compounds and hope to find something in nature that inhibits this enzyme. And so we developed an assay which is, you know, allows for such screening. This is a fluorescence-based assay, and it's quite simple. You know, the polymerase jumps onto this RNA, and then it extends this RNA, and as it moves to the right, it will displace a fluorescent probe, which has this yellow star dye here. And as you displace that probe, you discontinue to have a quenching with a neighboring fluorescent probe. And so you have a strong fluorescent signal as the polymerase goes to the end of the template. And this is an assay which allows you to monitor polymerase activity. And it's also an assay that can be used in a high throughput system, which allows you to screen thousands and thousands of drug candidates. So this assay works. We can see over time that the fluorescence increases when we add the active polymerase. And so now we could team up with Sonja Sievers and Herbert Waldmann at the Max Planck Institute in Dortmund. And they actually have this fantastic screening facility. It's a robotic screening facility, which will allow us to screen hundreds of thousands of chemical compounds and test whether they are able to inhibit the coronavirus polymerase. And hopefully we find something entirely new with a new principle. And that would be fantastic, because then we could check whether it also can reduce viral replication in cells and then later also in animals and so forth. So we are very hopeful that this works. And why are we hopeful? Well, I show you an example from a recent collaboration with the lab of Niels Göran Larsen. They work on the mitochondrial RNA polymerase and have done a lot of biology on it. And they have used this facility to find new inhibitors for it. And they found an inhibitor which we could then structurally localize within this mitochondrial polymerase, which is also a small polymerase, not like these big cellular machines. And this is actually an inhibitor that has a different principle and that is not a nucleoside analogon, but binds to an allosteric site. This is very exciting, and we will also use the mitochondrial polymerase system to look at side effects for the new inhibitors, because often these antivirals would also block the mitochondrial polymerase in our cells, which then leads to dramatic side effects. So here's a movie that shows you what we found in this collaboration with the Larsen lab. This was done by Hauke Hillen, who is a just amazing young person who now took on his own professorship and doing great things on mitochondrial gene expression. And you see in yellow here in space filling, you see the new inhibitor molecule, which actually has trapped a part of the active center of the mitochondrial polymerase, which is shown here in blue. This element is mobile, so the inhibitor blocks it in a different state. And this is a non-productive state, which then prevents the association of the mitochondrial polymerase with its natural DNA, RNA duplex that it has to bind for transcription. So you see here in blue the DNA, in red the RNA, and you see these clashes with the blue loop. So it's an allosteric inhibitor which rearranges the active site, and thereby it blocks the mitochondrial polymerase function. So this is our hope. We want to get something like that for the corona polymerase. Now, until there's good drugs, we of course need to go ahead with vaccination. And I can only tell you how excited I was when the mRNA vaccine was reported to work during the second half of last year. This was for me, one of the absolute highlights in my career. Of course, we were not involved in any of this. We just followed it. But, knowing the scientific background that enabled these biotech developments, going back to 1969 with the discovery of mRNA, then the genetic code and translation, all of this knowledge was important to design this mRNA vaccine, including, of course, modification of the RNA, which gives you then stability. So the RNA is not recognized as foreign and not degraded rapidly. It escapes the innate immune system. And even structure-based protein engineering to lock the protein that you want to express from this RNA into a functional state, which is then immunogenic. So this is a fantastic development of biotech industry based on decades of basic research, basic molecular biology. So how do these vaccines work? Well, you all know these mRNAs trigger the production of the spike protein in our cells, which is then secreted, and then the immune system recognizes this foreign protein. There's an immune response. And when we then later see the full virus, we will see the spike protein first. And since the immune system has been trained against this protein, there will be a rapid defense. And that's the principle of this active vaccination. So how does it work? How is it delivered? There's a nanoparticle that comprises the mRNA, which then can enter the cells. Then it's opened up. The mRNA is now accessible, and it will be translated by the ribosome. You produce the spike protein, and that is secreted, and then an immune reaction is triggered in cells. Now, when we go now to the DNA vector-based vaccines, it's a little bit more complicated. The principle is the same. You express an mRNA and then make the spike protein, but you have to go through the nucleus of cells. So you use this adenovirus, which is not harmful for humans, just as a vehicle to bring the DNA into the cell and into the nucleus. And there, this DNA vector is transcribed to make the messenger RNA. And then everything else is similar. You make the spike protein and so forth. And now I want to show you in the next 10 minutes how complicated this process is when the DNA has entered the nucleus and then is used for transcription and production of this mRNA, because this is actually the kind of research that we have been doing now for something like 25 years or so. And it involves this large cellular enzyme RNA polymerase II, which catalyzes the DNA-dependent mRNA production in cells. We live in this golden age where we have atomic structures of all 3 cellular polymerases, and including RNA polymerase II. And now we can move to very large assemblies and characterize them, because Pol2 is a very social enzyme, so it likes to associate with many other proteins. And very recently, and that is still unpublished, we managed to assemble the entire transcription initiation machinery for Pol2. And actually, this is now in the human system. We used to work in the yeast system, but now we went to the human system and we assembled these 15 proteins. It's over 2 megadalton, the whole complex. And it's interesting that we actually, a few years back, we decided to use adenovirus promoter DNA. So that is the promoter of the adenovirus used for vaccination. Also, people have actually now used even stronger promoters. But we use that one because it's known since 1973 to be a very strong Pol2 promoter. So we have now this whole complex and we can solve its structure. Again, we use cryo-electron microscopy. But I'm showing you here a picture of the European synchrotron in Grenoble to remind us that it's actually in the end a combination of electron microscopy and X-ray crystallography. Because over the decades, we and others have solved many, many structures of using crystallography of parts of this machinery, you know, 1 protein at a time, 2 proteins, 3, 4 proteins. And now we have this 50 subunit assembly, and we look at it by cryo-electron microscopy, but we can fit in all these smaller structures that we obtain from X-ray crystallography. And that actually results now in the first atomic model of the entire human transcription initiation machinery, which I'm showing you here now. And we build it up as we think it happens in nature. So you start with binding of the Tadder-box binding protein to DNA. There's a 90-degree bend introduced into DNA. And then 2 factors, TF2A and TF2B, would bind. And this allows you now to recruit the polymerase, which is here in silver, to the promoter, but only when TF2F is also present. This is the violet factor here. And now another factor, TF2E, joins and it binds TF2F above the DNA. So the DNA that has been loaded onto the polymerase is now retained on the polymerase. We call that the core initiation complex. Now a huge factor joins called TF2H, which is another 10 protein subunits. And it binds the downstream DNA. And you will see this is a very important protein because it has a motor domain inside of it. And then the so-called mediator complex, a large, in this case, 20 subunit complex will bind. It stabilizes the whole pre-initiation complex, but it also is stimulating enzymatic activities of the pre-initiation complex to get the polymerase started. Now, I don't have time to show you what we learned here, but I will just mention one aspect, and that is how the DNA is opening, because you need these proteins to open DNA, and the template strand needs to be available to start the production of RNA synthesis. Now, this process is very similar, believe it or not, in yeast and human. So you may think these 2 structures here are the same, but they're actually not. One is from yeast, the other is from human. This is how well the enzymes and the whole complex is conserved in evolution throughout all eukaryotes. Now we see in this movie how the DNA opens. It's again, you know, this technology where we have different structural snapshots and then we can actually animate the process and visualize the motion. So here is this motor, it's a so-called translocase. It's in red and it will use the energy of ATP hydrolysis to propel the DNA into the active site. And the first step is that it opens 5 base pairs of the DNA. So you just loosen them a little bit so that Watson-Crick base pairing is disrupted. And there's 2 loops in the polymerase that stabilize that situation. And then there's a second spontaneous step where this 5 base pair loop is extended to 15 nucleotides. And the template strand is then passing by the active site all the way here at the bottom of the enzyme. So there's a 2-step mechanism of opening. And we could even now visualize the details of the motor, how the motor works, how it actually moves the DNA in and how it opens the first few base pairs, how it disrupts the Watson-Crick base pairing. And in essence, what happens is that binding of this translocase motor introduces a twist in the DNA. And then the binding of ATP will propagate this twist towards the active site. And this strain is then released and propagated in order to open base pairs further upstream. Now, we have not only visualized this initiation process, but also many other steps of transcription, especially at the beginning, steps of transcription that happen at the beginning of genes that are transcribed. And so we serve the community by providing this structural insights and also complementary functional information on the transcription process, which I cannot share with you. But I want to share one last thing, and this is because it also has to happen to express this or make this mRNA from the adenovirus vector-based vaccine. This has been characterized before, and that is a second regulatory step in addition to initiation. And this step happens during early elongation, and it's called promoter proximal pausing. So it's a very strange mechanism where the polymerase, once it has started, doesn't get very far. It only transcribes for 50 base pairs and then it's paused and it sits there, and it can sit there for minutes. And it needs an activatory signal to then be released into the gene body. And so what we found through not only our structural biology, but also our functional genomics, where we analyze transcription regulation in living human cells, we found that this pausing of the polymerase can have a feedback to initiation, and it can actually limit the rate of initiation. So it can actually regulate the amount of mRNA that you produce. So those kind of mechanistic details are very important. If you want to produce a lot of mRNA in a short time, you need to understand these switches. Then you can engineer them and you can make stronger promoters. That involves a lot of knowledge from molecular biology. So I want to show you in the last movie how that looks now in living human cells. So what we did is we developed an approach that we call multi-omics. So we combine different protocols of functional genomics, and then we use kinetic mathematic modeling to extract kinetic parameters from the transcription process as it goes on over the genome in human cells. And what we could extract are 2 parameters. One is the frequency of initiation. So how often does the polymerase initiate at a certain human gene? And we also measured the duration of pausing. So how long does this pause polymerase stay there? Is it 1 minute, 4 minutes, and so forth? And through those studies, we learned that the pausing influences initiation and that it's this additional regulatory kinetic step in transcription, and that is summarized here. So each dot is now 1 human gene. And you can see when you inhibit the release from the pausing, you extend the pause duration, which is here the y-axis, so you get longer pause durations, but at the same time, the initiation frequency, so how often the polymerase starts, is down-regulated. And we call this elongating polymerase can pose a limit on how strongly the gene may be transcribed by limiting the frequency of initiation. That's it. I think in the future, we will more often combine structural biology, in vitro approaches with our in vivo approaches in living cells and genome-wide, and having the entire toolbox is very important. But what is also important is to have computational biology, because computational biology is providing the glue between those 2 in vitro and in vivo approaches. So one is bottom-up, the other is top-down, but you need the computational analysis and modeling to connect those two. Here are the take-home messages. Hopefully, I could convince you in these 40 minutes that RNA polymerases are important, not only for studying cancer or diabetes or immune, but also in the course of understanding the coronavirus and also in fighting the coronavirus. So coronavirus itself has a polymerase, and we could show how it functions. We could explain how a drug, remdesivir, is able to slow it down, but not to block it completely. And I think, these insights will facilitate the development of novel drug candidates. That is our hope. Then our studies of mitochondrial polymerase can help to then test new inhibitors for possible side effects. Do they also inhibit the human enzyme? And our studies of the human polymerase II system actually helps to understand how these vector-based vaccines express mRNA. And actually, it rationalizes a lot of the things that the companies do to get high expression. I didn't mention all of them. Finally, the phage polymerase, I didn't mention yet, it's super important. Imagine now we have to get hundreds of millions of people, actually billions of people vaccinated. So we need kilograms of messenger RNA. And how do you make this? You make it using an RNA polymerase. And this is actually from bacteriophages, T7 or related. And there's a lot of knowledge also to make very good polymerases as workhorses to produce these mRNA vaccines. And that brings me to the beginning of my career, because I remember in the 90s, I was reading papers from Tom Stites, who studied the T7 polymerase, was the first RNA polymerase to be studied in structural detail. And now to see that it probably helps to rescue the world, that is quite something. So I mentioned already the long list of past and present co-workers, and here's the list of the fantastic people who have been involved in the work that I showed today during this presentation. In the first row, you see those people who were involved in cellular transcription, and on the bottom here, those who additionally or exclusively worked on the coronavirus system. I'm very grateful also to our collaborators, Claudia Höbarten, and also Henning Urlaub, a mass spectroscopist who is essential for most of what we do. And with this, I would like to thank you all for your attention. Thanks again for giving me this medal. It's a great honor and pleasure, and thank you so much for having me. Hopefully, I could tell you a little bit about what we're up to at this time. Thank you.
Yeah, thank you very much, Patrick, for this inspiring and exciting talk. It's really been fantastic to see this roundup of how so many different aspects of the pandemic have entered into your work and how many steps forward you've been able to make with your group.
Thank you.
Thanks again, and I'll deliver the medal and the certificate to Göttingen soon.
Thank you very much. Thank you.